Welcome to Lecture 3. Lecture 3 and Lecture 4 together make up a series that I call Microbial Lifestyles. And so Lecture 3 is the first part of that. Lecture 4 will be the second part of that. What do I mean by microbial lifestyles? Well, I just want you to learn what microorganisms need to grow. What do you have to give them? What conditions do they need in order to grow and reproduce? And so what we're going to try to do is look at the variables that make up a microorganism's ecological niche. So what is ecological niche? Well, in the sense I'm using, it is all the different conditions an organism needs to thrive in its environment. So let me give you an example. Let's think about a bobcat. So there are bobcats living in the woods around, at least where I live in Oregon. And these are predators, so they eat other animals. They have to. If they don't have access to other animals, they will starve. They can't live by eating lettuce or eating peppers or eating seeds. Other animals can. A cow can eat grass, but a bobcat cannot. So a bobcat's ecological niche has to include access to smaller animals it can catch and eat. It also includes the temperatures outside. A bobcat can live in a fairly cold environment where the temperature sometimes gets well below freezing, but it couldn't live in an environment where the temperature stayed above 100 degrees every day. And it couldn't live in an environment where the temperature was minus 50 degrees every day. It has some optimal environment that it needs. A bobcat would need the right kind of cover, the right kind of plants around it to hunt in. You wouldn't see a bobcat necessarily hunting in the middle of a flat plain. I believe they have to ambush prey, so they have to have things to hide behind. So from this, you can probably tell that I don't know much about bobcats. That's okay. It's an example. Another example would be a plant. So you could imagine a plant. It has a different set of needs. A plant needs sunlight. It needs the right amount of sunlight. If it gets too much, it will get burned. If it gets too little, it will bleach. It needs water, and it needs the right amount of water. If it gets too much water, its roots will rot and it will die. If it doesn't get enough water, it will desiccate and not be able to grow. Plants need access to carbon dioxide. They need access to oxygen. It's also important to think about the soil they have, because that really determines what happens to their roots. So soil that drains well will dry out. Soil that doesn't drain will not. And some plants need that dry soil. Some plants need that never drying soil. But the key is they need what they need. And if they don't get it, they're not going to be able to grow. They're not going to be able to reproduce. And microorganisms are really the same way. They have a set of conditions they need in order to reproduce. And kind of the same way a plant is, a microorganism, it's stuck in its location. It can't get up and move 3 miles to a better place. A microorganism can't move more than a centimeter over the course of its life in many cases. And for most of them, nowhere near that distance. And so it's either going to grow where it is, or it won't grow if it doesn't have the right conditions. And if it doesn't have the right conditions, and it won't grow, well, then what happens? Well, there are a few different things that can happen. If it's temporary, that the conditions aren't right, maybe it will just go into stationary phase. But if it's long-term, it could actually die. So a microorganism with the wrong conditions can starve and die. Some microorganisms can go into a sort of dormancy that will allow them to survive in a dormant state until conditions improve, and we will see that in this lecture. What you will notice about the microbial lifestyles as you learn about them is that we talk about chemistry a lot. They're single-celled organisms, and so they are not as concerned with the flow of water across the ground or things like that. They're concerned with which molecules they can get and what they can do with those molecules. To some extent, you will see they do need water. Some of them need oxygen. Some of them need things that we can imagine needing. But to a great extent, we have to think about the chemistry that they require. And that can be daunting. But in order to really see the world as they see it, we have to see in their language, and that really is chemistry. And so, like this slide tells you, some of the concepts we will look at are going to be fairly simple. Like we can talk about the temperatures bacteria need. There are a set of vocabulary words to describe different temperature ranges microorganisms might require, and you'll see that when we get to it. But in other situations, when we think about their metabolism, that gets more in depth, and you really will need to do a deep dive into these slides. Take your time and understand each slide. You will be able to, and once you've gone through them, you will have a great idea not only of how bacteria live, but you will understand your own cells a lot better. So I urge you to be patient with these and really dive in as much as you have to understand them. And we will all come out the other side a little bit more knowledgeable. We start with a very big question. Why are microorganisms small? And no one knows. We can't ask microorganisms why they are so small, and even if we could, they wouldn't know. It's like asking us, why do we have two legs? Why do we have two legs? Nobody knows. But biologists answer these questions through the lens of evolution. We say, well, in the understanding of modern science, any living thing is the way it is because evolution has pushed it to be that way, because organisms with those traits were successful in the past and they were able to reproduce and have offspring with those traits. And so we can think about small size of microorganisms through the lens of evolution. So we have kind of three different evolutionary hypotheses, and they don't disagree with each other. They could all be true, or none of them could be true. So the first one is the ratio of surface area to volume, and I'll explain this in the next slide. Another is diffusion of signaling molecules, and I will again explain this in the next slide. The last one is reproduction rates. So small sizes mean that it doesn't take long for bacterial cells to grow big enough to divide. And once they divide, they've gone through an entire generation, and they can mutate as they go through that process. And in the process of mutation, they can introduce genetic variability into a population that allows evolution to happen. It allows natural selection to happen. So the small sizes allow them to reproduce quickly, exploit new niches, and evolve quickly. So let's get into these first two that I didn't explain. The first one is the surface area to volume ratio, or the ratio of surface area to volume. And so we first talk about a cell's volume. So like how much cytoplasm is in a cell. And this correlates to how much a cell weighs, so its mass, but also its demand for nutrients. How many enzymes does it have inside itself? How quickly does it need to make new proteins? That all correlates to its size to some extent. And so if we think of a sphere, the volume of a sphere is, 4/3 pi r cubed, where r is the radius of the sphere, or the radius of a cell. So let's keep that in mind. That's how we calculate the volume of a sphere, and the volume roughly correlates to the cell's demand for nutrients. Then surface area. What does surface area correlate to? Well, remember that any kind of cell gets nutrients across its membrane. Nutrients have to cross the cell membrane to come in, and waste products have to cross the cell membrane to go out. And in many cases, the cell surface area is going to control how quickly that can happen. It's going to limit how many transport proteins there can be. It's going to limit how much passive diffusion there can be. So the surface area of a cell determines how quickly nutrients can enter and how quickly waste can exit. So we can calculate the surface area of a sphere at 4 pi r squared. And again, r is the radius of the sphere. So what happens if we make r bigger? So if the cells were bigger, what would happen? And that's what we should look at. Here we can get the ratio of surface area to volume. And again, we can think of it as surface area is the ability to transport chemicals, volume is the need to transport chemicals. So a happy cell has a lot of ability to do what it needs to do. So a happy cell is going to want to maximize its surface area and minimize its volume. And if we look at the ratio of surface area to volume from the formulas on the preceding slide, this is what we get, 3 over r. So as r increases, this ratio is going to get smaller and smaller. As this number gets bigger, this whole ratio gets smaller. So bigger cells have less ability to bring in nutrients and get rid of waste products. So that's that's surface area and volume. We just think that a very small cell has more efficient ability to move the molecules it needs across the membrane. And human cells, we don't really need that because we have a blood supply constantly keeping our cells fed. And other things like that. And most of our cells don't need to swim around and rapidly divide. Most of our cells are not in the process of doing anything like that. So they can be a little bit bigger. But most of our cells their radius would be like 10 times, 20 times bigger than a bacterial cell, not like 100 times or 1,000 times bigger. The last one is diffusion. So how quickly can signaling molecules go from one end of the cell to the other? How quickly could a messenger RNA diffuse from one end of the cell to the other? And so here we could look at other physics stuff and say that the time required for a molecule to move a distance via diffusion is proportional to that distance squared. So if it takes a minute for a molecule to diffuse 1 centimeter, it would take 100 minutes to diffuse 10 centimeters. And that's what this is showing. If a cell gets 10 times bigger, it will take 100 times longer for molecules to diffuse across it. And so a bigger and bigger cell will need to solve a lot of problems related to getting materials from one part of the cell to the other. And yeah, this can be important for all of that. And this is why biologists are kind of amazed at things like motor neurons, which are enormous. And when I learned about motor neurons, my first question was, how do they get messenger RNA all the way to ribosomes at the distal end? How would proteins get to the distal end? How would anything get from one end to the other? Well, the answer is, if it's by diffusion, it won't. It could take, it's on the order of like a century for a molecule to diffuse a meter. So there must be other ways of making it happen, and our motor neurons certainly have those other ways. Okay, well that is enough for this first video. Next we're going to start looking at the factors that really define microbial lifestyle. I delayed us a little bit to talk about why they are so small, but that should be the first question we answer in any microbiology course.